Okay, so based on the poll, it looks like most of you have like a little bit of interest in forensics, which is great because it's kind of related to the lecture today. So here's the overview of what we're covering. Unfortunately, we don't have the time to go over everything in biotechnology, so I'm just going to go over what I think are some of the most important techniques to know for the USABO. So I asked how many of you are interested in forensics, and if you don't know what that is, it's the study that's centered around detecting crimes. And so if you've ever watched like documentaries or Criminal Minds, you would know that DNA fingerprinting and profiling are really useful pieces of evidence. So the first technique we're discussing, which is PCR, is very vital to DNA profiling. And I'll explain how in a little bit. So first, polymerase chain reaction is a really common lab technique that's used to amplify DNA sequences. So because DNA usually comes in really small quantities, makes it pretty hard to study. And PCR is very useful in amplifying that so you have enough to study nicely. And you might think this is the same as gene cloning, which I'm not going over today, but it's still important. But these two are different types of techniques. Gene cloning creates copies of entire genes using recombinant DNA and host organisms. PCR only produces specific DNA sequence copies within a gene and takes place outside of living organisms. So if you still don't understand, you can use this analogy here. So if gene cloning was like checking a book out of the library, PCR is like mass photocopying just a few pages. So over here, you have the three basic steps: denaturation, annealing, and extension. I'll go over them in the next slide with a diagram, but over here, take note that each one of them is dependent on a different temperature setting. Okay, so during denaturation, you heat the DNA to separate the two strands from each other. So you can see this blue strand right here is separated into two. And if you remember from the chapter on proteins, high heat is a factor that causes bonds to break. So the same thing happens with the bonds in DNA. During annealing, you cool the DNA so that the DNA polymerase can help primers bind to the separated DNA strands. On this diagram, the primers are the red lines. And finally, during the extension phase, the polymerase adds nucleotides to the 3' end of each primer. Over here, that's the dark blue arrows. And your end product is two newly duplicated DNA molecules formed from each of the single strands in the original sample. So the extension phase might sound familiar because it's really similar to the last steps in transcription and translation. You can repeat this cycle up to 40 times in this machine on the bottom right. It's called a thermal cycler, which controls the temperature. And the number of DNA molecules is going to increase exponentially with every cycle. So one sample can turn into millions in just a couple dozen cycles. All right, and here's another diagram. So you see the thermal cycler. These are the components that you need to carry out PCR. And here's just a repeat of the steps, one cycle of it. So a problem that you might think arises with PCR is that if you're heating the sample enough to separate the DNA strands, then wouldn't that cause the DNA polymerase to denature too, because it's an enzyme? So the solution to this is something we call Taq polymerase. It's the DNA polymerase that comes from T. aquaticus, which is a bacteria strain that lives in hot springs and hydrothermal vents. It's very heat stable and actually most active at high temperatures. So it takes care of the aforementioned problem, which is why in the previous slides, you saw that the last step takes place at a pretty high temperature as well. So PCR was first carried out in 1985 and has had a really major impact on biological research. So as you can tell from how it works, it has really great speed and specificity. For the USABO exam, you should understand the 3 basic steps and also why Taq polymerase is important. There might be questions where you're presented with a scenario and they'll ask you which technique is appropriate. So knowing what PCR does and how it works is really helpful. And you see a picture from Manny from Ice Age here because a really interesting application of PCR is that it amplified DNA from a 40,000-year-old frozen woolly mammoth. And I mentioned forensics before. That's because you can amplify DNA from the fingerprints, blood, or tissue that you find at crime scenes. And you can create enough of it to analyze with other techniques. PCR does come with limitations though, because if you have even a single error, it carries over to the billions of copies you end up making. And it's also very susceptible to contamination. It's also not a substitute for gene cloning because it's limited to only really small DNA fragments. And you have to know the exact sequence that you're copying because you need to choose the right primers. And after this lecture, you can check out the link at the bottom of the slide for an animated review of PCR. Okay, so the next technique that we're going to study is gel electrophoresis. So if you successfully amplify the DNA sample from the crime scene with PCR, the next step is to actually analyze it. And this is what gel electrophoresis is for. Okay, so like PCR, gel electrophoresis is relatively simple and common to use in labs. And actually, if you take the AP biology class, you get to perform this lab and pick out a suspect for a crime scenario. So this technique separates DNA or proteins according to their molecular size, electrical charge, or other physical properties. The most common is size, and this is what we're going to be discussing. So the image you see on the right here is agarose gel. Agarose is a polysaccharide that acts kind of like a sieve. And you should know that DNA molecules have an overall negative charge because of the phosphate groups in the backbone. So DNA is going to travel from a negative to positive electric field placed on 2 sides of the gel. The agarose fibers allow the shorter molecules to move farther than the longer ones, and the DNA fragments are going to naturally separate into bands. Okay, and here's the setup for gel electrophoresis. You have a tank here that's connected to a power supply that generates electrical current. The blue square is the agarose gel, and the blue bands here are the wells. They have DNA or protein samples in them, and once you turn the thing on, they're going to start moving. In this case, it's like down and towards the right. Okay, so here are the basic steps. You place the sample DNA into separate wells on a negatively charged end of the agarose gel. Then you're going to turn on the electrical current, which prompts the molecules to move towards the positive electrode because, like everyone knows, opposites attract. And finally, you turn off the current once the bands stop migrating, and then you add a DNA binding dye. If you don't add the dye, you won't be able to clearly see the separate bands. And I'm going to show a video that animates both PCR and gel electrophoresis. So give me 1 second to stop sharing.
A crime was committed and we want to find out who did it. We have 2 suspects. A strand of hair was found near the crime scene. We will use gel electrophoresis The strand of hair is placed into a test tube, that is placed into the PCR machine. A polymerase chain reaction, or PCR for short, is a technique that works like a molecular copy machine. The sample is heated in order to separate the DNA strands. The sample is cooled in order for the primers to anneal or attach. The process is repeated until a usable amount of DNA is created. Holes are created in the gel that serves as a reservoir to hold the DNA solution. Mixtures of different sizes of DNA fragments are loaded into a well in the gel. The force needed to draw the DNA through the gel is provided by electricity. A power supply takes the standard alternating current electricity available from the wall outlet and converts it into the one-way, direct current needed to set up an electrical field across the gel. The gel box is the container that holds the gel submerged in running buffer. It is designed so that when current is applied through the electrodes attached to the box, the current flows through the gel, creating the electrical field needed to push the negatively charged DNA molecules towards the positive electrode. Results are usually photographed in order to save the findings. Wells are then compared together for similarities. If two fingerprints match, they are probably from the same DNA source.
Okay, so that was the video. So here are what the results of PCR look like. The video didn't explain this clearly, but each of the samples are probably going to come from a different source. So one of them could be the fingerprint found at the crime scene, and then you could use fingerprints or DNA that was already in a criminal database. So if one of the ones from the database matches with the one from the scene, then it's pretty convincing evidence for who was at the scene. And it's not just DNA at crime scenes that this is useful for. Like I mentioned before, you can use this on proteins, which differ by size, charge, and other chemical properties because they have side chains. And instead of identifying a suspect, you can determine what kind of virus or plasmid you're looking at, or see which of your parents has a certain genotype. Okay. So the next technique we're looking at is called restriction fragment length polymorphism. So this is a technique that's closely related to gel electrophoresis. And first of all, you should know that a polymorphism is a variation in DNA among a population. And a restriction enzyme is an enzyme that cuts DNA when it recognizes specific sequences called restriction sites. So it's going to cut the DNA at those restriction sites. So a change in even one base pair in a sequence can prevent a restriction enzyme from cutting there. So that will leave you with one large fragment instead of two small ones. And we call the changes where this happens the restriction fragment length polymorphism. Gel electrophoresis separates by size. So if you have RFLP, it will result in a different band pattern than the original one. So here's an example that you've all probably heard of. It's sickle cell anemia. You know, I think we all watch like the same exact video on it in science class almost every year since 8th grade. So you should all know that this disease is caused by a mutation of a single nucleotide in the human beta globin gene, which also happens to be an RFLP. As you can see, there are three MST2 restriction sites on the normal gene. That's the one on the bottom, but in the mutated one on top, the restriction enzyme is only going to cut twice. because it doesn't recognize the one in the middle anymore. So the binding pattern becomes different. If you look at the one below number 2 on the pedigree, it doesn't have those 2 bottom bands. And so this is an easy way to identify if a person has sickle cell disease or not. And it works for a lot of other disorders too. Okay, and the last technique we're going over is called blotting. So there's a common expression that goes finding a needle in a haystack, which is kind of what this is about. If you have a mixture full of protein, carbohydrates, lipids, and nucleic acids, you want to be able to separate them or identify the ones you want to study. So this is where blotting comes in. You separate the molecules by size by running the sample through gel electrophoresis, and then you transfer the macromolecules onto blotting paper using capillary action to produce a pattern exactly like the one in the gel. So if you imagine putting a piece of paper on a fresh painting, pressing it on and then taking it off again, you're going to have something that looks very similar to the painting. And that's pretty much what happens here. You then expose this to a solution full of probes. Probes are complementary fragments that are tagged with some sort of color or light, maybe radioactivity. The probe molecules are going to bind to the macromolecule of interest. That's how you design them to be. And then now you have the macromolecules you want tagged with that same color and light. So the 3 main types of blotting to know are southern, northern, and western blotting. Each one corresponds to a different kind of macromolecule. And my favorite acronym to use for this is snowdrop. So basically you write the word snow and drop vertically next to each other, and the letters are going to line up with each other. So first one, S for southern, is corresponds to D for DNA. N is northern, lines up with R for RNA. O you can just ignore, and then you have W, western, matches with P for protein. All of these different types of blotting, they follow the same general principle, but each of them also has some few specific characteristics you probably want to look into. And there's great explanations for them in Campbell. And lastly, I'm going to leave you with some techniques in biotechnology that are great to study on your own. So know how cloning genes using bacterial plasmids and vectors work, and see the real life applications of them, how people have been trying to clone animals. You know, the most famous example is Dolly the sheep. I think it took like 277 attempts, and she was one of the only ones to survive. Cece the cat is another really good example. Its name stands for carbon copy. She was the first cloned pet, and it's really interesting because her fur color was a different color from the donor that she was cloned from. And this was due to some things like X inactivation and also maternal genes. Another one that's important is gene therapy. And even though it's a pretty controversial issues nowadays, it's still good to know. So know how people are trying to alter their genes inside people's bodies or for future germlines. People want to correct mutations that are causing diseases and disorders. And one of the recent trials has been with SCID, which is severe combined immunodeficiency. And they're trying to correct the mutation or change the gene sequence using retroviruses. If you're interested in this, I linked the PubMed article for those of you who like reading scientific literature. But this isn't required if you just want to take the exam. You know, there might be a chance that it shows up because USABO is like so specific and tests on literally anything, but it's still good to know if you just have a passion for biology in general. And something that affects all our lives, of course, is genetically modified organisms. There's a lot of those in the crops that we consume as food every day. Okay, so I have a couple questions from past USABO exams. So they were too long to fit onto a Kahoot or Mentimeter. So I'm just gonna have you guys message into the chat what you think the answer is. And I'll give you about 1 minute to do this problem. Okay, so here's the answer. It was A, you're not going to see any bands. And that's because this is actually a trick question. So if you read the first sentence, it says you obtained red blood cells from an individual, and then you ran a DNA gel electrophoresis on it. So the thing is, red blood cells don't have nucleuses, so they won't have DNA in them. Normally, you would see two different bands for heterozygous because you have two different alleles, and one for homozygous. But for this question, none of it really applies because you're looking at red blood cells. And I have one more question. This is from the 2012 USABO, I think. And I'll read it. So you perform a Western blot on two proteins of similar molecular weight, and you find that only one band developed on your SDS page gel. So how would you modify your assay to distinguish the two proteins? Okay, so this one was a really tricky question because it requires you to know some things about this specific type of gel. So first of all, the answer was C, focusing your sample isoelectrically on a pH gradient. So this method is based on the fact that proteins have specific charges at certain pH values. The acidic and basic functional groups on the amino acids increase or decrease the total charge of the protein, right? So you need a special gradient gel or that's what it's called here. And because there's an electric charge connected to the gel, the protein travels to the point in the gel where the charge of the gel equals that of the protein and the total charge equals zero. That's why it's called an isoelectric point, where there's an equal amount of negative and positive charge on the molecule. So as for A is wrong because even though you want a higher resolution, which would, oh, sorry, I forgot to explain. So the reason that only one band developed was probably because the resolution was too low or you didn't have enough separation between the bands. So there's like a difference, but you can't see it clear enough. So the goal is to do something that makes the bands separate more. So as for A, you want a higher resolution, but to do that, you want a higher concentration of acrylamide, not a lower one. It's a chemical substance that you can use to make the gel, like quote unquote, harder. So the harder the gel is, the more it restricts the movement of larger proteins while still allowing the small ones to pass through. So if you're restricting the big ones and not restricting the little ones, then they're going to separate more and you're likely going to have two bands. So A would be correct if it said higher instead of lower. So for B is also wrong because even though you want to denature your proteins, you want to denature your proteins so that they're all a constant linear shape. But ionic detergents are much preferred over the non-ionic ones because the non-ionic detergents have a pretty limited ability to break the bonds that aren't covalent. And they also result in a non-uniform charge throughout the molecule, which is something you don't want. Okay, so D, you definitely don't want to remove reducing agents because reducing agents help with the denaturation of the protein. They break the intermolecular disulfide bonds. So removing the reducing agents is not going to help at all. And lastly, switching the cathode and anode won't do anything. The proteins are still going to migrate in the direction of the opposite charge. So C is the right answer because it helps separate the molecules, the proteins even more based on their charge. And the other ones I'll have either just don't do anything or are the opposite of what you want. Okay. And that's about it. At the end, I linked a Khan Academy practice for biotechnology. So if you want more problems, they're a lot easier than the USABO ones. But if you want more practice, here's where to get it. Yeah, and that's all I have for today. I think they all left.